Hi, my name is Carrie, and I'm going to be talking about the ABO blood group system. And so I will get started on that, and y'all can follow in this Word document with me and take your own notes or just listen. Get that pulled up. So we're going to be talking about our ABO blood group system, and where you get those from is they are antigens defined. Antigens defined by immunodominant sugars on the cell surface. And specifically here in a minute we're going to talk about which sugars go with which antigen, but first I need to give you all a little overview about the H antigen. Your H antigen is going to be your precursor structure where A and B antigens can be made. So precursor structure, where A antigens can be made. It is found on chromosome 19 with your secretor gene. So the H antigen that is found on chromosome 19, which happens to be where the secretor gene is also located, and your ABO inherited chromosome or inherited chromosomes on chromosome 9. So not always a test question, but extra information. H is not part of the ABO blood group system. However, it must be inherited to form the ABO antigens on the red blood cells. So it's not part of the system, but it's needed to form it. Must be inherited to form ABO antigens on red blood cells. And similarly, the secretor gene I kind of touched upon earlier has to be inherited to form ABO in the secretions. So H is for the red blood cells, and secretor gene is for your secretions. And now I'm going to try to make this just a little more comprehensible. So your H antigen is your precursor for all of your blood types, but it's specifically if you just have H antigen, then that's going to type is type O. So pretty much your O blood group is the same, or your O blood type is the same as your H antigen. So I think this picture will help a little bit. So you've got your precursor, which is this H antigen, and if you have the H antigen, then that can go into one of three groups. It can go into a group with A, so you can have A antigen and H antigen. You can have group B for B antigen and H antigen, or you can have group O, which is only having the H antigen. And that is why O is a universal donor, because it does not have that A and B, but both A and B antigens are also accompanied by the H antigen. So again, if it sees that H antigen, it sees the type O blood as its own self in most cases. Um, However, O does not have the A or B, and so it'll make antibodies for both. Now, if you don't have the H precursor antigen, then you will have what is little h, and that is going to be your Bombay phenotype. And in that case, there's a very limited number of potential donors that you could have, and so it's not the coolest thing to get, but comes up in blood bank once in a while. So I think that made it a little easier. Um, Now your sugars responsible for specificity. We'll start with H, now that you know what H is. So H specificity is going to be with the L-fucose sugar. Your A specificity is going to be with N, and it's going to be acetylgalactosamine. And then your B specificity is going to be your D galactose. Now, whatever way you can remember something is always the best way. I'll give you a couple for me. I've had students tell me that the letters starting them are generally close to each other in the alphabet. So like the H and L are close to each other in the alphabet. And then for myself, I didn't have one so much for the H and L, but the A for A specificity also starts with your acetylgalactosamine, and then your B goes with D. They kind of sound alike, they kind of look alike, and I always remember like the BD tubes, the manufacturer BD, and so those always kind of worked in my brain together. And now, as I mentioned earlier, you have the special type that is Bombay. Bombay. And what happens there is no H antigen, which means it lacks normal expression of ABH, or it could also be ABO, or the equivalent of ABO antigen. Be written as O with a little h. Let me see if I can make this look right. It can also be written as little h, little h. Oops, and I want to keep that little h. Now, getting into a little more ABO, they are your ABH antigens are constructed on oligosaccharide chains. Again, not always a test question, but sometimes helpful. So they are, let me put that up here. Constructed, no, I'll put it under. oligosaccharide. So ABO in general, so we got ABO antigen. They are generally strong reactions. What that's referring to is about 3+, 4+ reactions when you're running your forward and reverse typing. Um, they are generally at immediate spin, so your and if you don't know, immediate spin is going to be your room temperature, which is about 20 to 24 degrees Celsius. Now, if you know that, generally you can figure out what kind of immunoglobulin it is, and so your room temperature or your initial spin immunoglobulins (Igs) are generally IgM, and then your IgG is going to be your 37 degrees, which is generally your clinically significant antibodies because they react at body temperature at 37. But these are going to be generally IgM. They are inherited predominantly, which means you get one from mom, one from dad. ABO antigens can also activate complement. And if you've learned so far, IgM and IgG are both efficient complement activators. IgM specifically because there's so many binding sites. Okay, now there is some basic information that I really think helps whenever you're doing forward and reverse or anything to do with ABO typing, and comes up when you get a lot of questions on this. You might know how to do it in the lab, but it's really good to help know all the terminologies and where you'd find them. So I'm going to start with antigen. So antigen is going to be found on the surface of red blood cells. So that can either come from reagent red blood cells or from a patient or donor red blood cell. And then antibody is going to be found in sera. So either anti-sera, reagent anti-sera, or in the serum from the patient or donor. Next, you've got reagent red blood cells. They will always be red and blood-like color. And again, if you can't remember, that's going to be your antigen. And then you've got reagent, anti-sera. It's going to be clear or colored. And when I mean colored, it's going to be dyed. And it's going to consist of your antibody. And now I want to point out that whenever we're talking about reagent, this is going to be a known amount of substance, whether it's a known antigen or a known antibody. Anything reagent will be known, and anything patient or donor will be unknown. So known is going to be reagent, and unknown is going to be your patient or donor sample. Patient or donor sample. And when you are determining the antigen, that's going to be your forward type. If you think about it, your forwards like your big kahuna, and your antigen is what you're talking about when we're talking about ABO and antigen. And then your determining antibody is going to be your reverse typing. Let me see if I can. Still working on my tech savviness. Okay, so now what you need for each one of these for the reverse typing is one of each. So you need one known and one unknown. You also need one red blood cell product and one sera product. So either reagent anti-sera or serum from a human. And then you also need one antigen and one antibody. So you need either your reagent, grade red blood cells or anti-sera, and then you need either whatever the opposite of that is, patient serum or red blood cells, and then you will in turn get one antigen and antibody out of that. And I want to note whenever, oops, whenever you are working with red blood cells, whether it be the patient's or your reagent, RBCs inverted and properly mixed. So if you just invert it a couple times and look at the bottom of your reagent red blood cells, that should be a smooth color all the way through. Now, a couple more little helpful tips is going to be, so we talked about known and unknown. So you have your known, which is going to be, and tell me if you know, going to be reagent. And then I pre-made this chart, I'm sorry. Let's see. I'll do it this way. Okay, so your known reagent is going to be either your red blood cells, antigen, or your anti-sera, like your anti-A and B, which are colored yellow and blue. It's going to be your antibody. And another nice little chart is going to be your, we started there with known, so we're going to start with unknown, which is going to be your patient or donor sample. Because you'll see this abbreviation a lot, so know that the PT is patient or donor sample. And then that is going to branch off into either your patient or donor red blood cells, which is what? Antigen, or patient or donor serum, which is where the antibodies are floating around. There we go. There we go. So we got known reagent, unknown patient or donor samples, and now you know which samples can be used for either one. And then I'm going to do one more little chart. And again, I just think these are really helpful when I think they're really helpful when you are getting started to have something like this to point you in the right direction, because it gets confusing for some people. We've got in vivo, which is where? In the body or in the lab? In the body, and then in vitro is going to be in the lab. And so in the body, you can have antigen or antibody. So I put that out as, let's see, there's again. So we got antigen, and So what's your sample going to be in the body for antigen? It's going to be patient or donor, red blood cells, and then for antibody, what is going to be your in vivo sample for antibody? Be your patient or donor serum. And if you wanted to make another chart for in vitro, the reason I didn't do that is because in vitro you're going to use both. So you're going to use antigen from both red blood cells, patient or donor red blood cells, and from reagent red blood cells, and you're going to use patient and donor serum and reagent anti-serum. Now, once you have these down, you can start with your forward typing. And I think I still got space. So, forward typing. What is forward typing? So it's going to be your big guy, your antigen. It determines if an individual possesses certain antigens. What is that going to equivalent to? So we know that we're talking about an individual. So that's going to be your patient or donor. And then we're going to be talking about antigens. So it's going to be red blood cells. So it's going to be, we're going to be talking about our patient or donor RBCs, which is the equivalent of your unknown antigen. And so I'm going to put, because if it's patient or donor, we've got unknown. And since we know it's RBCs, it's going to equate to unknown antigen. And then forward grouping or typing is defined as using known sources of commercial anti-sera to detect antigens on an individual's RBCs. And see, all of this is just different terminology for the same thing. The anti-sera that you're generally going to use is your anti-A and anti-B, which again are clear, but they dye them yellow and blue to help prevent them getting mixed up with any other anti-sera. Blood leak is so important to make sure that you don't make mistakes because it can get deadly really quickly. So we talked about the anti-sera that we're using, but how do you figure out, first of all, what you need to start with? So like I said, if you have your unknown antigen right here, then you know you need the opposite of that. So the opposite of an unknown antigen, opposite of unknown, is of course going to be known. And then the opposite of antigen is going to be the antibody. So you know that you need to start with a known antibody. And so if you look up at the chart up here, what's known, so known is going to be a reagent. So here are two choices. Going to be your reagent red blood cells or your anti-sera. And now we know we need a known antibody. So our antibody is going to be anti-sera. So we know that we are going to start with reagent anti-sera, which happens to be anti-A and anti generally used ones. And then you need to mix that. Mix with, and now you need, we talked about needing up here one known and one unknown, one red blood cell product, one sera product, one antigen, and one antibody. Let's see what we got. We have reagent anti-sera. So we've got one known. So we need one unknown. So we know we're going to mix it with the patient or donor. So we got one known and one unknown now. And now we've got, we don't have a red blood cell product, but we've got one sera product. So we know we need a red blood cell product. So we're going to mix it with patient or donor RBCs. And then we've got one antigen and one antibody. So we've got our antibody, we've got our antigen. So we're going to take reagent anti-sera. Oops. And I just remind myself every time I read or write like reagent anti-sera, clear or colored. And then every time I think about patient or donor red blood cells or red blood cells, I think about them being red. And so that way you can picture it in your head when you're doing your labs that you have the correct material that you're using. So for forward typing, we're going to take our reagent anti-sera and mix it with our patient or donor red blood cells, which is our unknown antigen. Now we need to know what we are looking for. So looking for, we're also testing for. So we know we are looking and testing for antigen. And anytime you're looking or testing for, that's going to make it an unknown. So we know we're looking for the unknown antigen, which equates to your patient or donor red blood cells. So whenever you get asked a question about forward typing, it determines if an individual possesses certain antigens. So you're looking or testing for your unknown antigen, and by using known sources of commercial anti-sera to detect those antigens on an individual's red blood cells. Now I'm going to insert our result chart for that. The forward is pretty straightforward and rather easy. It is going to follow exactly what your results are, which is always nice. So if you have a positive for anti-A, it's going to be group A. Positive for anti-A and negative for anti-B is going to be group A. Negative for anti-A and positive for anti-B is going to be group B. And then positive for both A and B is going to be a mix of both A and B cells, so they will be group AB. And then if you are negative for reagent anti-A and anti-B, then you will have group O cells. Now we're going to do reverse typing. Determines an individual has certain, you know what we're looking for? Antigen or antibodies. Can be certain antibodies. And so this is going to equate to an individual and has certain antibodies. So it's going to be your patient or donor is your individual, and your antibodies is going to be your serum. And so your patient or donor is going to relate to unknown, and your serum is antibodies. And I know you could jump straight to antibodies, but I think it helps to go through the whole process and go over it every single time. Helps you remember a little better. So it's going to be unknown here. Antibodies. And then we got a reverse grouping or back typing is defined as detecting as detecting ABO red blood cells. I'm sorry, ABO antibodies in the patient's serum. Defined as detecting ABO antibodies in the patient's serum by using known reagent red blood cells, which is generally going to be your, I'll put it down here, but it's going to be your A1 and B cells. And again, those are going to be red. Okay, so we're going to start. I'm trying to make them like equivalent to each other. So when the notes come out, they're looking the same. So we're going to start with, we know that this is unknown antibody. So the opposite of that is going to be known. And now the opposite of antibody is going to be your antigen. So what is our known antigen? So if we come up here and we go to our chart and we've got our known, and we find the better known. There we go. So our known antigen is going to be here. Here's known. And we've got our antigen, which is going to be our reagent red blood cells. Known for reagent, and our antigen is going to be red blood cells, which is your A1. And B cells. And those are going to be red. just like the blood. And so what's important, if they are a blood cell product, you need to be mixing them properly and make sure that they are thoroughly mixed in an appropriate way. So we've got our known antigen. So you know we start with our reagent red blood cells, and so we need to mix those with something, right? So mix with. So now we need to know that we have one known, one unknown, one red blood cell, one sera, one antigen, and one antibody. So we've got our reagent, which is known. So we need our unknown, so we know we need our patient or donor. And now we've got our antigen, so we need an antibody. And so we know we're going to need some kind of sera, and we're referring to our patient or donor. So you go up here and look for your patient or donor. And if we are looking for antibody, then we know we're going to need patient or donor serum. And that is going to be our antibody. Reagent red blood cells. We'll mix that with patient or donor serum. And now what we're looking for So what are we looking for? We are going to be looking for unknown antibodies. And now I will put in on the results chart. It's going to be the opposite of your forward typing. So your if your A1 cells are negative and your B cells are positive, then you will have the antibody in your serum will be anti-B, because you can make an antibody against the antigens that you do not have. So it'll be anti-B, and so you will interpret as group A, because you're not going to make antibodies for your own blood group. So if you're group A, you have the A antigen, and you are not going to make antibodies for A. So if you have antibodies for B, that means that you do not have the B antigen and can make antibodies against them. So that's kind of how that goes backwards. So if you're positive for A1 cell, then negative for B cell, then you have the anti-A antibody, and you type as group B. If they are both negative, then you have no antibody in your serum, which means you express all those antigens, so you are group AB. If you are positive for both A1 and B, it means that you have both antibodies in your serum, and you are type group O.